Welcome to session 3 of Principles of Inheritance and Variation. In this session, we will be seeing about inheritance of two genes, in which we will be seeing a dihybrid cross. We will be seeing about law of independent assortment, chromosomal theory of inheritance, and linkage and recombination. So, talking about inheritance of two genes. Mendel also worked with and crossed pea plants that differed two characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape and one that has green colored and wrinkled shape. Mendel found out that the seeds resulting from the cross of the parents had yellow colored and round colored shaped seeds. So here you can tell which of the characters in the pairs, yellow or green colored and round or wrinkled shape, was dominant. What was dominant? Yellow was dominant. Round was also dominant. Okay, so thus yellow color was dominant over green and round shape was dominant over wrinkled. These results were identical to those and he got, and when he made separate monohybrid cross between the yellow and the green seeded plants and between the round and wrinkled seeded plants. So it was similar to the result when a monohybrid cross was performed. So now let us use the genotypic symbols. Capital Y for dominant yellow seed color and small y for recessive green seed color. Capital R for round seed shapes and small r for wrinkled seed shapes. The genotype of the parents can be written as capital R, capital Y, and small r, small y, small y. The cross between the two parents can be written down as in figure 5.7, showing the genotypes of the parent plants. The gametes are capital R, capital Y, and small y unite on fertilization to produce the F1 hybrid capital R, capital Y, small y. So when Mendel had self-hybridized the F1 plants, he found that 3/4 of the F2 plants had yellow seeds and 1/4 had green seeds. Okay, so the yellow and the green seed the yellow and the green color segregated in a 3:1 ratio. So round and wrinkled seed shape also segregated in a 3:1 ratio, just like a monohybrid cross. So we have the image over here. So what can we see? We have the P generation. We have round yellow, wrinkled green, which gives round yellow. Okay, and but the genotype changes. You can see that. So the F1 generation on selfing, what it gives is such a table. So this is the Punnett square. Okay, so it gives 9:3:3:1 ratio round. So here is the ratio. Okay, round yellow, round green, wrinkled yellow, wrinkled green, 9:3:3:1. Excellent. Okay. So now we will be seeing about law of independent assortment. What does this law state? So in a dihybrid cross, the phenotypes round yellow is to wrinkle yellow, round green, and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. So the ratio 9:3:3:1 can be derived as a combination series of three yellow, one green with 3 round and 1 wrinkled. So this derivation can be written as follows: 3 round is to 1 wrinkled into 3 yellow is to 1 green is equal to 9 round 9 round yellow, 3 wrinkled yellow, 3 round green, and 1 wrinkled green. So based upon such observations on dihybrid crosses between the plants differing in two traits, Mendel proposed a second set of generalization that we call the Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of character. So which is the very, very important law. The law states that when two pairs of traits are combined in a hybrid, when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of characters. Just think about the law, and when you write it, automatically the words will flow. So when two pair of traits are combined in a hybrid, that is very important, are combined in a hybrid. When two pair of traits are combined in a hybrid, segregation, okay, that is also a very important word. Segregation of one pair of character is independent of other pair of characters. Okay, so the Punnett square can be effectively used to understand the independent segregation of two pairs and of genes during meiosis and the production of eggs and pollens in the F1. Okay, so what they say is independent segregation of two pairs during meiosis and the production of eggs and pollens in the F1 capital R, small r, capital Y, small y plant. Consider the segregation of one pair of genes capital R and small r. 50% of the gamete have the gene capital R and the other 50% have small r. So now besides each gamete having capital R or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of 50% of capital R and 50% of small r is independent from the segregation of 50% of capital Y and 50% of small y. So therefore 50% of the small r bearing the gamete has capital Y and the other 50% has small y. Similarly, 50% of the capital R bearing gametes has Y, okay, and the other 50% has small y. Thus there are four genotypes of gametes, four types of pollen and four types of eggs. Okay, so the four types are such a type, right, with a frequency of 25% or 1/4 of the total gametes produced. When you write down the four types of eggs and pollens on the two sides of a Punnett square, it is very easy to derive the composition of the zygotes that give rise to the F2. Although there are 16 squares, how many different types of genotype and phenotypes are formed? Note down the given format. So there are only 4 types of phenotypes and genotypes we have to count. Totally there are 10 different types of genotype, right? So basically everything except these 6. All these are different from each other. You have to note that. Okay. Chromosomal theory of inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till the 1900s. So firstly, communication was not easy as it is now. In those days, and his work could not be widely published. So number one is communication. Okay, and secondly, his concept of genes or factors in Mendel's words as stable and discrete units that control the expression of traits and of the pairs which did not blend. With each other was not accepted by his contemporaries as an explanation for the apparent continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. So finally, though Mendel's work suggested that factors genes were discrete units, he could not prove any physical proof for the existence of factors or say what they were made of. So in 1900s, three scientists, De Vries, Correns and von Teschemark. De Vries, Correns and von Teschemark. Okay, remember this for God's sake. Very important scientists. I don't want to forget them, right? So independently, they rediscovered Mendel's lesson on the inheritance of character. Also, by this time, due to the advancement in the microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of the structures of the nucleus that appear to double and divide just before each cell division. These were called chromosomes. Chromosomes are colored bodies. Okay. So they could see the nucleus. They could discover the structure of nucleus that appear to double and divide just before cell division through microscopy. Okay, so by 1902, the chromosome movement during meiosis had been worked out, and Walter Sutton and Theodore Boveri, they both noted that behavior of chromosomes were parallel to the behavior of genes. Okay, so behavior of chromosomes were parallel to the behavior of genes. And he used chromosomal movement to explain Mendel laws. Okay, so recall that you have studied the behavior of chromosome during mitosis, equational division, and during meiosis, reduction division. Okay, the important things to remember are that chromosomes as well as genes occur in pairs. So the two alleles of a gene pair are located on the homologous sites on homologous chromosomes. Homologous sites on homologous chromosomes. Okay. Now moving on. So this is the meiosis and germ cell formation in a cell with 4 chromosomes. Okay. So now can you see how the chromosomes segregate when the germ cells are formed? So this is G1. Okay. There are 4 different chromosomes. Then they become bivalent. Then meiosis takes place. Then now they become 2 different chromosomes in each thing. Okay, and then again 2 different chromosomes in a single. So this is a reductional division because 4 chromosomes are there, and it becomes to 2 chromosomes only, and then again 2 chromosome is being maintained. So this is an equational division. So from this we find out that Mendel's law can also be explained with the chromosomal theory of inheritance. It need not be alleles, that has to be genes that has to be ensured. So they found out that the similarity in the behavior of the chromosome and the genes. So basically A is genes and B is chromosomes. Okay, so now during anaphase of meiosis 1, the chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of 4 different colors, the left and right column. Okay, so during anaphase of meiosis 1, the 2 chromosome pairs, okay, I have to show you the whole thing. Anaphase of meiosis 1. Okay, the 2 chromosome pairs can align the metaphase plate independently of each other. And to understand this, compare the chromosome of 4 different colors in the left and right columns. In the left column, possibility orange and green is segregating together, but in the right column, possibility 2, the orange chromosome is segregating with the red chromosome. Okay. So one long orange and one short green chromosome, and one long yellow and one short red. But over here, one long orange and one long red. So the red is been together. Orange green is together. Orange green is not together. Red. It is red. So how we obtain different germ cells? Okay, this can be observed. So I hope it is very clear in the mind. Okay, so now we come to our most favorite part, the chromosomal theory. So Walter Sutton and Theodore Boveri, they argued that the pairing and separation of pair of chromosomes would lead to the segregation of a pair of factors they carried. So the separation of a pair of chromosomes would lead to the segregation of pair of factors they carried. So Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Okay. So, following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan. Thomas Hunt Morgan, very important. Okay, what he and his colleagues, they led to the discovery of discovering the basis for the variation that sexual reproduction produce. So Morgan, what he did, he worked with the tiny fruit flies, the Drosophila melanogaster. What is it? Drosophila melanogaster. So basically we say Drosophila, we have heard it so many times, right, when ma'am took genetics class. So Drosophila melanogaster, very important, please remember. Okay, which were found very suitable for such studies. These could be grown on simple synthetic medium in the laboratory, and they complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and the female flies are easily distinguishable. Also, it has many types of hereditary variation that can be seen with low power microscopes. Right? So Drosophila melanogaster, they could be grown in the laboratory. Point number 1, complete their life cycle in 2 weeks. Single mating, large number of progeny. Okay, and there was clear differentiation between the sexes, and many types of hereditary variations that can be seen with low power microscope. So very 6 important points, 5 at least you can see, right? Lab 2 weeks, large number of progeny, sexes differentiable, and then low power microscope, the hereditary variations could be seen. Coming to our most favorite topic, linkage and recombination. So this is one of the most interesting topics of genetics. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex linked. So the crosses were similar to the dihybrid cross carried out by Mendel in peace. For example, Morgan hybridized yellow-bodied white female to brown-bodied red males, yellow-bodied white females to brown-bodied red-eyed females, and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3:3:1 ratio expected when the two genes are independent. So Morgan and his group knew that the genes were located on the X chromosome and saw that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of the parental gene combination were much higher than the non-parental type. Okay, so he saw that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination were much higher than the non-parental type. So Morgan attributed this, okay, this was the MCQ last time. Okay, Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on the chromosome, and the term recombination to describe the generation of the non-parental gene combination. So Morgan and his groups also found that even when genes were grouped on the same chromosome, some genes were very tightly linked. Okay, so some genes were very tightly linked and showed only 1:3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Stutwart used the frequency of recombination between the gene pairs on the same chromosome as a measure of the distance. So he found that the frequency of recombination between gene pairs was used as a measure of distance between the genes and mapped their positions on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of human genome sequencing project described later. Okay, so this so what did Alfred find out? Alfred Stutwart, Morgan student, he found out that distance was inversely proportional to recombination. Okay, now I will teach you this process. This is a very easy process. Okay, now so now we have cross A, cross B, parental F1 generation. Okay, so we can see we have yellow white wild type. Okay, so yellow white was crossed with red and brown, right? So Morgan hybridized the yellow bodied white eyed female to the brown bodied red eye male. Okay, so this was a wild type, and of course we know that this doesn't carry the Y chromosome does not carry the genes, only the X chromosome does. So this denotes the Y chromosome. What happens? There is a what happens in the F1 generation? This goes with this and this goes with this. So what happens? This goes. So orange, blue, you find that out, right? Yes, and this is dominant. Okay, now first of all, after you get this, then okay, basically this goes with this, this goes with this, and this goes with this. Now this and this and this and this, sorry, this and this and this and this, right? Okay, and okay, first of all, this cross, okay, and then this cross, this and this combined, this and this combined, you can see. Then this and this combined, this and this combined, you can see over here, right? Now again, for recombination, what happens? Y plus, right? Y plus, and then W, it combines with this Y plus, and then Y, then W plus and Y. Okay, it combines here. Then the exact opposite, then same Y plus and W plus, Y plus and W over here you can see, right? And Y and W plus, you can see here, it combines with the other chromosome. So very simple, right? Again, coming to cross B, so you can see that this is a different cross. What happens? So over here the genes are together, very closely linked, tightly linked. Over here they are separated well. Okay, so distance is inversely proportional. No, distance is proportional to recombination. I was wrong last time. So greater the distance, more recombination takes place. Okay, so we can see this, we can see this, we can see this. Same thing takes place. The same type of cross, that's all. Okay, so we are done with okay, so figure 5.11, I would like to read out a few things. So linkage is the result of two dihybrid crosses conducted by Morgan. Cross A shows crossing between G, Y and W, right? Gene Y and W, and cross B shows crossing between gene W and M. So here dominant wild type alleles are represented with positive sign in superscript, and the strength of linkage between Y and W is higher than W and M. So linkage is inversely proportional to distance. Meet you in session 4.